Welcome to Provitalks, where nature and science blend to inspire you. Discover the latest in technology, sustainability, and trends in the cosmetics world, thanks to Provital. Let's care together for our industry's commitment to the people and the planet. Provital. Do care.
Welcome to a new and very special edition of Provitalks, an edition specifically dedicated to dive into the state-of-the-art in vitro experimentation in the world of cosmetics research and development. The thing is that every day more often, the cosmetics market demands advanced technological developments that will open new horizons in cosmetic science while reducing time consumption, cost, and environmental footprint of the techniques used to such developments. Today, and thanks to our expert, Dr. Miguel Aso, we will discover the opportunities that the most advanced in vitro techniques can offer in cosmetics experimentation. Welcome, Miguel.
Hello, thanks for having me.
Thanks to you. We believe that with your study and research of skin tissue during your years at NYU, your experience as an in vitro lab manager at Provital, you will be able to inspire many of the professionals in the sector who listen to this podcast. So thank you very much for coming here today, directly from the laboratory, to enlighten us with your knowledge.
Thank you very much, Clara. It is a pleasure to talk with you about our work in the in vitro lab. By the way, these Provitalks are a very interesting initiative. I believe that popularizing science is a part that researchers usually forget, but it is essential if we want the non-specialist public not to get carried away by trends without a scientific basis and to understand the real progress of scientific advances.
Of course. Miguel, do you mind telling us a bit about your experience as an academic researcher before entering the field of cosmetics?
After obtaining my doctorate in pharmacology, I moved to New York to join the NYU, New York University, Department of Translational Medicine. There, I focused my efforts on understanding the intracellular signaling mechanisms responsible for collagen synthesis and its role in scar formation. In other words, in some way, I was already in the field of dermocosmetics during my academic days.
Yeah.
The point is that this training allowed me to know in detail the cellular processes of the skin, which is being of great help to me now to develop in vitro tests and strategies for the study of Provital's active ingredients. The central role of the in vitro laboratory is to design robust assays to understand the mechanisms of action of active ingredients. However, at Provital, we are very clear that an in vitro laboratory of a cosmetic company cannot focus exclusively on this aspect, but rather has to combine the robustness of the test, which is in itself a major challenge, with being continuously navigating the latest scientific advances. Thus, from Provital's in vitro laboratory, we have been acquiring internal skills in everything that has to do with big data applied to the study of the skin. I'll try to go deeper into this later.
Of course, surely our listeners will love delving into this concept of big data that we've heard so much about it lately. But first of all, let's recap a bit. In a previous Provitalk, Blanca Martinez, our then R&D manager, mentioned the immense advances that we have been seeing in artificial skin models since the first ones that came out as an alternative to animal tests. Could you tell us more about it? How is the optimization of this technique contributing to the scientific or technological development of private companies? Could you tell us about any application of these skin models in the process of developing a cosmetic ingredient?
Indeed. As Blanca told you, ethically and as it is logical, the field of cosmetics should not experiment on animals. This is a big difference with respect to the pharmaceutical industry, where animal experimentation is justified from my point of view, since the purpose of this industry, unlike ours, is the cure and treatment of diseases. That said, this ethical ban, which prevents us from testing cosmetic products on, say, mice, has been one of the best examples of pressure for innovation. I'll explain. During my time at the NYU, I did work with mice. There we studied scar formation and tested active molecules that could potentially improve the healing process. But as I have mentioned, in the field of cosmetics, the use of animals is not ethically accepted. In fact, the European Cosmetic Regulation prohibited it in 2013. This caused researchers in dermocosmetic science to make an extra effort, which would never have been done without this ethical prohibition, which has led us to the development of reconstructed skin models. I would like to explain the true meaning of these models. I have the impression that the information that the non-specialist reader interested in cosmetics has about these reconstructed skin models is that they are models that are halfway between animals and human volunteers and cell cultures. And it is true, but I will add a point that I only realized when I started working with these models. And that is that before, when I worked with animals, we had two very different approaches, very differentiated. On the one hand, we tried to delineate the molecular mechanisms through what we call intracellular signaling pathways responsible for the action of active molecules through specific cell culture techniques. While on the other hand, to go on to study the effects on the whole organism as a better reflection of the biology in humans, we tested these molecules in mice. One of the most important points in studying whole organisms is that you can see, literally see, what is going on in the skin, specifically in a slice of real skin where you see the different layers of the cells. To do this, samples of the entire skin are taken and histologies are performed, which essentially means staining the skin and then observing it under the microscope, because in the end, seeing is believing.
Yeah.
And this has always been beyond the reach of cell culture assays. Well, when developing the reconstructed skin techniques here at Provital, I realized that one of the most beautiful virtues of these models is that even though they are basically cell cultures, albeit in 3 dimensions, keratinocytes, for example, are actually being differentiated into the different layers of the epidermis. And you can even observe how cells change in shape until they become, in the outermost layer, corneocytes. And it is tremendously interesting that we can apply staining techniques that we could previously only use on animal or human explants now on in vitro cultures. Mm-hmm. This is really a game changer.
Yeah.
In this sense, at Provital, we are proud to have developed the stretch mark reconstructed skin model. Through this internal development of Provital, which we published in the prestigious scientific journal, International Journal of Cosmetic Science, we were able to replicate at the molecular level and at the structure of the extracellular matrix the defects found in the stretch mark skin. We used this model, once it was well defined, to study whether our active ingredient striover was capable of improving stretch marks and by what mechanism. Indeed, we found that striover improves stretch mark in terms of both molecular markers and looking at the structure of the skin under the microscope. To our knowledge, this is the first time this model has been successfully shown, and we are very proud of it. And not only that, at Provital, we are betting on these 3D skin models. And proof of this is that in collaboration with the Autonomous University of Barcelona, we have a predoctoral researcher, our colleague Julia Barcer, doing an industrial doctoral thesis focused precisely on these skin models.
Yeah. How could you not be proud of these techniques? Well, Miguel, going back to big data, you said before that Provital is also using the most advanced technologies for the study of skin biology, where the application of bioinformatic tools, statistics, and machine learning methods is a prerequisite for current biomedical database research. How can computational analysis of large data sets in high-throughput experiments help open new ways and targets in skin biological studies?
It is true what you say, Clara. When I started my research career 18 years ago, gosh, time flies so fast. I remember that the bulk of experimental work was dedicated to what in scientific jargon is known as wet lab. That is, the work we all have in mind when we talk about a laboratory, pipettes, cuvettes, petri dishes, reagents, etc. Then, of course, some time had to be spent analyzing the data using specific software programs depending on the techniques used. But little by little, the work of a researcher is changing towards dry labs. This is research based on biological data, but carried out with computers, which are dedicated to data science, so that we now perform fewer experiments, but from which much more data is obtained, millions of data. Now, the complex part is the subsequent analysis of the data. This requires knowledge not only of specialized programs, but directly of programming language, which has a very steep learning curve, but which once certain knowledge has been overcome, it is a tremendously powerful tool for massively analyzing data.
Yeah.
If I had to choose a single idea to summarize the impact of big data in cosmetic science from an in vitro point of view, I would say that going from measuring some parameters to measuring all of them, we find answers to questions that we had not formulated to begin with. And this gives us a more complete picture of the effects of our active ingredients in the skin, and it allows us to detect interesting effects that we had not even considered investigating. This has already provided us with powerful breakthroughs in discoveries about our ingredients.
Well, what an impact. It is really incredible what an industry like the cosmetics can achieve thanks to the application of these techniques of analysis and interpretation of results. Thank you very much, Miguel, for this first podcast specifically dedicated to the most intrinsic part of the development of cosmetic active ingredients, the part that is normally less known by the final consumer. So I hope our listeners have found this layer of the beauty world as interesting as I have. To you, I say thank you all very much for listening. And if you found this podcast interesting, you cannot miss our next Provital with Miguel. Thanks, Miguel, and see you all next time.
Thanks to you. I'm looking forward to talk to you again.
Thank you for listening to this edition of Provitalks. We hope you got some source of inspiration to create and spread your own kind of beauty. For further information, subscribe to our podcast or visit our website at weareprovital.com. Provital. Do care.